Welcome to the de.NBI, ELIXIR Germany, and GHGA Knowledge Series. Today, we have a webinar with Wolfgang Maier, and the topic is Galaxy for Analysis of Human Health Data. Maybe I can introduce the organizers. So de.NBI and GHGA, this project. So the webinar series is a collaboration between de.NBI. de.NBI is the German network for bioinformatics infrastructure, and it is the German node to the European initiative ELIXIR. As you see in the map, de.NBI is a huge network with a lot of different universities and research data centers, and covering diverse topics and specific expertise in various bioinformatics topics. So from human or plant bioinformatics over microbial informatics to systems biology, and maybe the main facts about de.NBI. de.NBI provides, develop and maintain a lot of tools and services. And they also have a huge training offering in really diverse topics. And they also deliver computer resources for academia in Germany. So they have the de.NBI Cloud that we at GHGA also use. And and they have a lot of collaborations nationally and also internationally via ELIXIR. And one of these collaborations is this webinar with GHGA. GHGA, this is German Human Genome-Phenome Archive. We are a national secure omics data infrastructure, and we provide fair and controlled access to data while ensuring data security. So we are working always in compliance to the GDPR and other laws. And we are an NFDI consortium built of 6 data hubs across Germany and coordinated at the DKFZ. And we have different collaborations nationally and internationally. And at the international level, we are the German node to the European initiatives such as the European Genetic Data Infrastructure and the Federated European Genetic Archive. And well, we have also a lot of training offerings. And if you want to access the training or learn more about us, you can visit our webpage. The same, I forgot to say the same, is for de.NBI. And now the most important thing, I want to introduce our speaker today. Today we have the pleasure to have Wolfgang Maier. Wolfgang Maier made his PhD in molecular neurochemistry in the Max Planck Institute for Brain Research and Biophysics in Frankfurt. Then he was a postdoctoral researcher in genetics at the Friedrich Miescher Institute in Basel, and currently he's lecturer in molecular genetics and bioinformatics at the University of Freiburg, and he is also a member of the Galaxy Europe team in Freiburg since 2018. Yeah, 2018. So welcome, Wolfgang. It's a pleasure for us to having you. Thank you very much. And now the floor is yours.
Thank you very much for the introduction, Vanessa. Thank you for having me here. Thanks for the invitation. Maybe small add-ons. So within the Galaxy Europe team, I'm overseeing our portfolio of genomics, genetics, workflows, pipelines, tools, and everything surrounding that, so related functionality. And yeah, I will reserve the first couple of slides today for a more general picture of what Galaxy is and what it's useful for. So for people who really don't know much about Galaxy yet, this will be interesting. For the remaining audience, the main focus will be on new features that I selected because I think they're really interesting for analyzing human health data specifically. Although Galaxy, of course, is not that focused platform, so you can really analyze data from all kinds of sources. It's completely data agnostic, so to speak. So let's get started with the introductory slides. And so, yeah, at its very core, Galaxy is a web-based platform for giving users graphical, operating system independent, controlled access to whatever compute you decide to attach to it as the administrator, and to whatever storage you attach to it as well, for performing really any kind of data analysis. And the code for this Galaxy Core project is free and open source, lives on GitHub as shown here, and is developed by a large community of contributors, but specifically by core groups in the United States and in Europe and in Australia. So Penn State University is one of the important contributors on the US side, and then Freiburg in Germany also as a de.NBI node is one of the important contributors on the European side. And then there's the Australians that have distributed their efforts more all over the continent. If you decide to run your own Galaxy server, which you can by just cloning that code from GitHub and then launching it, you would get a web interface very similar to what's seen here. So this is a screenshot from Galaxy Europe, but your own version of it that you could host yourself wouldn't look that much different. And in general, you have analysis tools that you can install into this web interface, into your Galaxy, and that your users can use to analyze the data. That would be on the left side. You have a middle panel that you can use to announce all kinds of things like we do on Galaxy Europe with news and events here. And then you have a history panel, and this is where the data that users produce accumulates and where they can like have an overview over all the analysis that they've been doing so far. Now, as I said, as an admin, you can install tools into this server, and we're also doing this on EU, obviously. For this, you don't need any installation skills, so you don't have to have any specific knowledge about the tools you're trying to install and what requirements they have, but you're relying on containers or conda packages, and you can install those together with Galaxy wrappers that instruct Galaxy how to create a visual graphical user interface around these tools. You can install them from this kind of app store that we call the toolshed. So, and this is then inside Galaxy in a special admin section. You can just graphically with a one click on install or uninstall manage the installation of tools. And this is aware of different versions of these tools. So you can have several versions side by side and users can then pick which ones they want to use. So this is just how you would populate a freshly set up Galaxy instance with analysis tools. And then, yeah, on the user side, this presents itself like this. So when a user selects a tool from the tools panel on the left side, they can see the graphical tool interface that's defined by this Galaxy wrapper of the tool they selected. And then a few things that are important to note here. So when you click on these stacked boxes icon here, that's the different versions of a tool that are installed on the server. So there's full reproducibility here. You can always go back and use a previous version of a tool that you've used for previous analysis to get the same results out of it again. There is server-side reference data like the human reference genome, for example, in this case, version HG38, that a Galaxy admin can also install using graphical user interfaces onto that Galaxy. And we have probably thousands of reference genomes on Galaxy Europe that you can pick from. Yeah, and that makes the whole thing very convenient for the user. They select the tool, the version they want populated with parameters, choose reference data that the tool might need, and without any installation hassle, they can just run that tool and get their analysis done. Another strong aspect about Galaxy is the training material. So there's training.galaxyproject.org. I have at the end a slide with all the links collected. So that is a collection of currently close to 500 different tutorials that show various scientific data analysis conducted with Galaxy, introductions to the Galaxy user interface, to managing your data with Galaxy, and also like technical development tutorials for getting tools into Galaxy and whatnot. So you really have a lot of information there. We are very grateful for having this resource developed by the community and funded and supported by various partners. And for example, you could, if you want to learn how cut and run data analysis is done on Galaxy, then you can follow this tutorial. And the nice thing about it is that all the steps that are described in the hands-on boxes, so the stuff that you're actually running as part of the analysis, will just work on major public Galaxy instances or any Galaxy instance that has those tools installed. So there is no extra effort by the user. They just populate the tool interface of the respective tools like shown in the tutorial, and then they can follow this entire analysis. Speaking of public platforms. So there's Galaxy Europe, there's Galaxy, usegalaxy.org, the original main US platform. There's usegalaxy.org.australia, and then other servers like usegalaxy France, for example, that are many of those are freely accessible to the public. You need to register for an account, but otherwise you are free to use them. And those public instances often have many thousands of tools installed, as you can see here, and a wide selection of reference genomes for really any kind of data analysis you might be thinking of. What Galaxy is on a somewhat bigger scale is not just this core platform that you clone from GitHub, but it's the platform plus the compute and storage infrastructure that you as an admin decide to install or attach to it. Then the tools you're installing, the reference data you're providing to your users, and finally that associated training material that's shared between the different instances. And together, that's like the classical Galaxy ecosystem, I would say. But as I said, what I want to focus on today is recent developments around Galaxy. And as I said, I've picked some that I hope are interesting for people working with human health data. So these are mostly from the last year, some from the last 2 or 3 years. And even people having used Galaxy recently might be surprised about how things have changed. So might be worthwhile to present a few of those. So first of all, workflows and the Intergalactic Workflow Commission workflow library. You might not yet have encountered this, but Galaxy now has a peer-reviewed workflows repository. Think of it as similar to NF Core maybe for Nextflow workflows, from all kinds of scientific domains, but also for human genetics, epigenetics, genomics, whatever. Data analysis. So according to the screenshot, we have currently 109 such peer-reviewed workflows from all kinds of domains. And you can filter them here on this web page and then decide to run any of these workflows directly in your preferred public instance of Galaxy, like Galaxy Europe, for example. There have been lots of recent enhancements around workflows in Galaxy, and one of those that I love most is the workflow invocations tab, reachable from the activity bar here on the left side of the Galaxy instance. And this is really giving you a list where you can zoom into the details of every prior workflow run that you did under your account on this Galaxy server. And it will show you like the analysis steps conducted, the inputs to flow run, the parameters you've configured for this workflow run, which steps succeeded, which might have failed. So here they are all green, so completely successful workflow invocation. And there are several interesting things hidden here at the top of this interface. So the workflow invocation is connected to a data analysis history. So you can directly jump to the data that was produced during this workflow run and then inspect the results. You can run the same workflow again with the run button. You can also decide to rerun this specific workflow invocation, which will pre-configure the same inputs and parameter settings again. Yeah, and then on these tabs here, so we're currently looking at the overview tab, but you could switch to the export tab, for example, and then you can export the entire workflow invocation if you wish, complete with resulting datasets as a workflow run or crate, for example, functionality that we've implemented together with ELIXIR partners. Or as a biocompute object, or as a simple compressed file. You can also say, I only want the steps exported, but not the data produced by them. There's a metrics tab, which lets you investigate which is the computationally most expensive step of your workflow, or which are the most computationally intensive steps of that workflow. Like here you can see that BWA-MEM, for example, in this particular invocation, took a combined allocated core time of 128 seconds in that case, but it's not the dominant factor in this workflow. But that's like good for figuring out bottlenecks where you could maybe enhance your workflow, make it more efficient. So that's really nice overview if you're a heavy workflow user. And together, all these enhancements really make workflows now like the core, I would say personally, the core feature of Galaxy. So that's a very satisfying situation there. And it's maybe a bit similar, but probably more than what you're getting with things like Sequera Tower, so in the Nextflow world. So you can really manage all the analysis you've ever done and jump between data back and forth. Yeah, and this, I mean, we're only at the beginning of this. I mean, this has been improved over the last years, and now we're seeing a ramp up in workflow executions that people are running on public servers. So this, for example, is the Galaxy Europe statistics. So overall on Galaxy Europe, we had 660,000 and something workflow executions so far. But this increases swiftly now with all these new features. Then I want to switch topics a bit to interactive data exploration, because historically that's always the thing that people had a bit trouble with on Galaxy. You have these tools that you run, and then they run on some machine in the attached compute cluster. You're getting results back. But if you want to explore data, people have often complained that this is a bit cumbersome to do on Galaxy and has a lot of waiting time in between and guesswork. And this has improved drastically over the last couple of years. I want to pick single cell data example for this to illustrate the current situation. And that's because there's the single cell lab page, and that's the address is here. And it's really an outstanding example of community effort, in particular by the ELIXIR single cell community. And also a very nice example of such a Galaxy Labs page. And as a small excursion, if you want to learn how you can give your own community a landing page and an analysis platform, a tailor-made analysis platform in Galaxy. Then, of course, the Galaxy training material. What else has information about this? So you can go to the section on Galaxy community building and you learn how to build such labs pages and so on. The single cell labs page has tons of useful information like how to import public single cell data onto Galaxy, resources recommended for this, links out to workflows that the community has created that you can directly use on this page. So this is using infrastructure from Galaxy Europe, but has its own selection of tools and its own landing page. That's how these lab pages work. Yeah, and this community has done some fantastic work. So one is that they implemented interactive exploration of the structure of single cell data objects. Like one of the widely used data formats in the single cell world is undata format. And here you can see interactive exploration of the data structure of one such undata object dataset. So you can really go into all the attributes that this dataset has encoded and like the raw data inside of it and inspect, display all of it. So that's quite helpful to understanding what you're dealing with, specifically if you downloaded such a dataset from a public resource. But then there's also now integration of some super cool interactive tools for single cell data analysis, one of them being the cell by gene tool. So this is based on the official cell by gene tool from GitHub. And that's like you can open these interactive tools directly from the Galaxy user interface and start them with datasets that you have in your current Galaxy history. And then you can really graphically explore all kinds of aspects of your single cell data, see which genes are expressed in certain clusters of cells more than in others, correlate this to genotypes, cell types, whatever you like, and then even export some of this data back into your Galaxy history, like differentially regulated gene sets that you found. You can just export them into your Galaxy history, leave the cell by gene interactive tool and continue with your analysis where you left off. So that's really, really powerful. And yeah, speaking of public data sets, for some of them, we have really very convenient direct integration into Galaxy as removed file sources. So one of them is the tabular sapiens data that is now available to all users of Galaxy Europe. So you can just go to upload, choose from repository, and then tabular atlases will be one of your options there. And then you can select any of the undata data sets from different tissues, cell types, import them into Galaxy, visualize them with the cell by gene interactive tool and start your own analysis from there, or use it to template analyses you're thinking of. Good. Another area where big external data is super useful is machine learning. That's a field where it's often notoriously difficult for users to set up certain software themselves because they need to download models, train them, and so on. And that might be very hard for non-absolute experts. And there's one cool new tool that I want to highlight here, and that's Flexinesis, a deep learning toolkit for multi-omics data integration and getting conclusions out of that data. This was developed by de.NBI partners at the Max Delbrück Center in Berlin. And it has first-class full Galaxy support and integration through tools and training material. So the training material has 4 different tutorials currently about flexinesis-based analysis. And what flexinesis does is it takes multi-omics data from, well, different sources. Yeah, it could be like transcriptomics data, genomics data, special omics data, whatever you like. And then you're setting as a user just some simple parameters where you're saying what you want to, which aspects of this data you want to have analyzed. And then it can, depending on the data you're trying to analyze, it will pick the right deep learning model for the task. And you can say whether you want to have classification, clustering, regression analysis, and so forth. And this is an excellent tool for getting a first idea about data and the information contained in it. Or to, after you already learned something about your data, to confirm that you haven't missed anything that might still be there to discover in it. Yeah, one thing you don't have to care about as a Galaxy user is, of course, installation, shown in this image here. So that's all done for you. The models are there. You can, or the software is there. You can train your models and just start analyzing. And this is a screenshot what it's like to work with. So in this case, this is one of the tutorial data sets for flexinesis from the original publication about glioblastoma cells. And here the tool learns to classify high risk and low risk glioblastoma cases. And it also learns which genes protect or are risk factors. So mutations in which genes protect or are risk factors for glioblastoma associated mortality. And it correctly identifies here one of the protective mutations that are known in glioblastoma. So this is really, you just give it all your multi-omics data and it basically tells you like a summary of what's in the data. And then you can still take a deep dive and analyze more precisely certain aspects. And yeah, speaking of this deep dive, of course, we don't only have like these specific tools like flexinesis or cell by gene, but there's also as an interactive tool, you nowadays have Jupyter-based notebooks in Galaxy. So at any point in your analysis, you can also just import within Galaxy where you start a Jupyter notebook and within Galaxy you import data from your history into this running Jupyter notebook, do your data analysis, data exploration there, and you can also export results back into your history from there. Then the final aspect, maybe the most interesting one that has changed a lot in the Galaxy world, is the whole data in, data out story. So where does external data come from? How does it get into Galaxy and where can it be stored? There have been huge developments over the last 2 years in this area. So one is if you now go in a modern Galaxy instance like Galaxy Europe to your user preferences, you will find an option to manage your repositories. And what that does is it gives you the option to connect any from a huge variety of cloud or remote file sources to your Galaxy user account. So whatever an institution or an organization gives you access to in terms of remote storage, you can connect to Galaxy and you can say, I have access to this or that Nextcloud via the WebDAV protocol. I have S3 storage somewhere. I have a Zenodo account or whatever. You can connect it to your Galaxy user account. And when you do, you can say, I want to have this readable or writable or both. And then if it's readable, you can again go to choose from repository in the upload manager, and you will find these repositories that you've configured there in the list of choices. So like I've configured my Zenodo account there. I have a Nextcloud account at the Department of Computer Science. I've configured that. So these are my choices. And then I can go into any of these and get my data from there directly into Galaxy. And there's also an export datasets tool there in the tools panel, and this makes use of those same removed file sources. So if they are writable, I can export any of my history data to such a removed file source by selecting the source here. And then, for example, in this case, that's all my available export options on Galaxy Europe, and I could export my data to any of these remote places. And the file and possibly index files, if it's a BAM or CRAM file, I can export there. And this is usable in workflows. So you can write a workflow in Galaxy that at the end just dumps key results files that you're expecting from this workflow onto some network share that you have access to. So that's a really elegant way to get your data out of Galaxy again. And then what you also have is manage your Galaxy storage. This is a related but slightly different concept. So here you're saying I have some remote storage and I want to have Galaxy use that and manage it for me. So Galaxy will create a complete object store there and really use it to store all the data sets from your history there if you want to. So again, you have a couple of choices. So any kind of cloud storage that you could think of is available. So S3 compatible storage, for example, Google Cloud Storage, more specifically Amazon S3 storage if you have access to it. You can select whatever you want, give your credentials, and then for example, I'm having a S3 bucket from our university managed via tenant that I've configured for my account. And then what you can do in a workflow run, for example, you can say, okay, this workflow is producing special kind of data, for example, human health data, right? And that's why I don't want to have it stored on the Galaxy instance, but I rather want it to be stored somewhere else. So Galaxy will still create a history with the resulting data sets, but the underlying data will actually go directly to the storage for this particular workflow run that you're selecting here. So I can say all results of this workflow should please go to my university S3 storage. And so as long as that storage is connected to Galaxy, I will then have it available in my Galaxy history, can do further computation on it. But when I say I decouple the storage from Galaxy, then I can still see that I've produced some data from my history. I can see that, but I will not have access to the underlying data anymore. So this has two aspects. So first of all, it's like this your own storage that you're bringing with you. It doesn't count towards your user quota. And the second aspect is you stay in full control of your data and you can basically just pull the plug and say, okay, I don't want this data accessible through Galaxy anymore. What's interesting is that you can combine this functionality with the deferred dataset functionality. So this is again in the upload dialog. If you have some data that you're going to import into Galaxy from a remote repository, then you can, in the settings for this upload, you can configure that dataset resolution should be deferred for this dataset. And what this will mean is that Galaxy won't ingest that input data into its object store. So it will just note where the data lives and then pass it on to a tool doing computation on it. And the results will be stored wherever you said it should be stored. But the input data will never be ingested into the Galaxy object store and will just live remotely. So in that case, you can configure Galaxy basically to not store your input data. And if you bring your own storage, you can also instruct it to just store all the data there, and none of that will ever touch Galaxy's default internal object store. Yeah, and we're currently working on extending this further. So currently, if you really have sensitive clinical data, then your best option is to just have an in-house Galaxy server that's running um, um, behind your own firewall and so on, and you would just do your analysis there. But where we are slowly getting towards is that you can use any server basically, and you feed data in, you get data out, but the data really is not touching that server concretely. And so you can switch out different analysis options, Galaxy servers, also for the sensitive data. What we're exploring there is using Crypt4GH standard at the moment, so that Galaxy will not even have access to metadata about your samples, because currently in your history, it stores things like a small glimpse into your data, like the first few kilobytes of data can be previewed, and that's stored in the Galaxy database. Number of variants in a VCF file, for example, is stored in the database. Reference, genome, these things could be stored, and potentially those could be sensitive as well. And through the use of Crypt4GH, we would like hide all this information from the Galaxy server completely, and when combined with deferred datasets and remote object stores, that could give a nice very privacy-oriented flavor of Galaxy. So that's work in progress, but all this stuff already works. Yeah, and that's the end of my brief tour of selected new features. There's plenty more, so feel free to ask lots of questions. I'm looking forward to them. Here's a couple of useful links for you to the Galaxy Hub that documents everything going on in the Galaxy community and in the project globally. Galaxy technical documentation, training material, the help forum, Galaxy Europe that I showed most examples on now. And then if you want to keep up to speed with what's going on, I mean, we're having regular news posts on the Galaxy Hub. If you want to have an automated feed for those, consider Mastodon. So there's a Mastodon account of either the Galaxy project globally, and the US team also uses that, or if you want to follow the Freiburg team, so the team behind Galaxy Europe, then the other link is yours, the Bavü social one. Yeah, and with that, I thank you all for attention, and yeah, hope there are good questions.
I want to say thank you again. Thank you to our attendees, to our participants, and thank you to Wolfgang for the talk. And of course, thank you to my helpers and the other co-organizers, Daniel and Julia, and the other people from GHGA. And yeah, if you have future questions, you can reach out to us. And yeah, and thank you very much, and have a nice day, I will say.
Thank you very much.